Hello everyone, welcome to my channel Biology with Madhuparna. Today I will be discussing with you about the glycolysis. This video will be about the overview of glycolysis, and in my further future videos I will be discussing about the two phases of glycolysis, preparatory phase and payoff phase. So from the scratch we will start, like from the very beginning we will start, like what is glycolysis we will know, and what are the things which is important for national exams like GATE, NET, ICMR, DBT, all these things. So let's start. First, we will be looking into the definition of glycolysis. So, what is glycolysis? Glycolysis is the breakdown of glucose by enzymes releasing energy and pyruvate. It is the first step in the breakdown of glucose to extract energy for cellular metabolism. So, why it is the first step? So, if you look into glucose metabolism as a whole, you can see first a glucose molecule, it will go to glycolysis, then after that from there it will generate pyruvate, and then that pyruvate will be converted to acetyl CoA, and that acetyl CoA will go to Krebs cycle, and further from there we will get NADH and FADH2, which will go to ETC and generate ATP, and which means we will get the energy, and which is required for our cellular activities and all these things. So next we will look at some important facts about the glucose. So as we are talking about the glucose metabolism, so we will be looking into some facts which are important. So the first fact is the complete oxidation of glucose to CO2 and H2O proceeds with a standard free energy change of minus 2840 kilojoule per mole. So whenever you are seeing this minus, that denotes that the overall reaction is spontaneous in a way. So our next fact is it is also a remarkably versatile precursor capable of supplying a huge array of metabolic intermediates for biosynthetic reactions. As we learn further, we will get to know that many of these intermediates of this glycolysis method, it will go to another biosynthetic reactions and play major roles. So that we will be seeing on other metabolic pathways. Next fact is, in animal and vascular plants, glucose has four major fates. So after one more fact it is remaining. So after this fact we will be discussing about what are the fates or major parts of the glucose and its utilization. So our last fact is organisms that do not have access to glucose from other sources must make it. So what are the examples of that? Like what are the organisms and how they make this glucose, that we will just study in a little bit so we can have a better idea. So photosynthetic organisms make glucose by first reducing the atmospheric CO2 to triose. Triose means a carbon precursors which have 3 carbon. Then what they do, they convert this triose to glucose, and then further they will be using this glucose via glycolysis to generate pyruvate and further ATP. Next is the non-photosynthetic cells make glucose from simpler 3 and 4 carbon precursors by the process of gluconeogenesis, which is effectively reversing glycolysis in a pathway that uses many of the glycolytic enzymes. So again, like one future video will be there which will be about gluconeogenesis. There we will see it is actually a reversible pathway of the glycolysis pathway, but with some exceptions, some reactions which are irreversible in this glycolysis pathway, those reactions will be replaced by other reactions in gluconeogenesis pathway. So we will come to our next topic, which is major parts or fates of glucose and its utilization. So if you look into glucose has 4 main fates or 4 major parts. So what are those parts? First and the most important is oxidation via glycolysis, which will generate pyruvate. Second one is glucose can also act as a storage. Glucose can be stored inside the body as glycogen, starch or sucrose. Third one is synthesis of structural polymer, where it is used for extracellular matrix and cell wall polysaccharides. For the cell wall structures, this glucose can be used. And the last one is oxidation via pentose phosphate pathway, which will lead to the production of ribose 5-phosphate, which will further go to the nucleoside, which will further help in nucleotide synthesis in our body. Next, we will talk about some of the important facts about the glycolysis. So before reading the glycolysis as a whole, we will read some facts which will be helpful. First is glycolysis was the first metabolic pathway to be elucidated and is probably the best understood. And this glycolysis pathway was discovered by Otto Warburg and Hans von Ellwanger-Chelpin in Yeast, and Gustav Emden and Otto Meyerhoff in muscle. So, in most organisms, glycolysis occurs in the cytosol. So, always remember, if this questions comes, many times I have seen this questions comes in your college exams, tests, and even sometimes in national exams also. So, where this glycolysis actually takes place? So, the answer is always cytosol. So, you should remember this one. Next, the most common type of glycolysis is the Emden-Meyerhoff-Parnas pathway, which is EMP pathway. And then glycolysis also refers to other pathways such as Enter-Dioderov pathway. Mostly this Enter-Dioderov pathway you can see mostly in gram-negative bacteria, certain gram-positive bacteria, and archaebacteria. So these were some of the important facts about our glycolysis. So we know that glycolysis have two stages. One is preparatory phase and another one is payoff phase. In preparatory phase, mainly we invest the energy, like as in ATP, we invest the ATP, and in payoff phase we get it back. So it's like, stock market. We will be investing some of the amount and we will get it back as a double or triple, like that. So now we will have an overview of this full glycolysis pathway, where we will talk about like how glucose is finally converted into pyruvate via 10 reactions. So let's start. So in the very first reaction, glucose is converted into glucose 6-phosphate with the help of the enzyme hexokinase. What this hexokinase does, one ATP will be coming. From that ATP, this hexokinase will take the phosphate group. It will attach to this phosphate group in the 6th carbon of glucose, and it will release the ADP back into the cytosol, and as a result glucose 6-phosphate will be generated. As it is attaching this phosphate in the 6th carbon of glucose, so its name is glucose 6-phosphate. In the second reaction, and this first reaction, it is the first priming reaction. Okay. Now we will come to the second reaction, where glucose 6-phosphate is getting converted into fructose 6-phosphate with the help of the enzyme phosphohexose isomerase. So just from glucose it is getting converted into fructose, so nothing else. So now we will come to a third reaction, which is one of the most important reactions. Okay. So, and if you look into overall glycolysis pathway, there are 3 reactions which are irreversible. That is first reaction, third reaction, and the last reaction, where we are generating the pyruvate. So these 3 steps are irreversible. Rest other 7 steps, it can be reverted back. So let's continue. So in the third reaction, fructose 6-phosphate is getting converted into fructose 1,6-bisphosphate with the help of the enzyme phosphofructokinase 1. So again, phosphofructokinase 1, what it is doing here, it is taking the phosphate group from the ATP, it is attaching that phosphate group to the first carbon of the fructose. So it is named as fructose 1,6-bisphosphate. As already in 6th carbon there was a phosphate, so now again it have attached one more in first carbon, so it is fructose 1,6-bisphosphate. And this is the pacemaker step of the glycolysis. Pacemaker in the sense, if a patient's heart is not working properly, then doctor will attach one small instrument, pacemaker, which will help your heart to pump the blood and like to go on. So this is the step where you can regulate this overall pathway. You can stop. So about that we will be discussing in our future lectures. So now we will be coming to the fourth step. In this step, fructose 1,6-bisphosphate is getting converted into two different products. What are those products? That is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate with the help of the enzyme aldolase. So till now we were dealing with one hexose sugar. Now we are having two triose sugars in our hand. So we will be coming to the next reaction now. So in next reaction, dihydroxyacetone phosphate, which is a previous product of our previous reaction, so that will be converted into glyceraldehyde 3-phosphate with the help of the enzyme triose phosphate isomerase. So now we are having two molecules of glyceraldehyde 3-phosphate. As you can see in the screen, I have numbered all the carbons like 1 to 6. So from the next reaction, as both now we are dealing with same molecule, both same molecule, so we will be only taking into count that 1 to 3, like first glyceraldehyde 3-phosphate molecule. So now we will come to our next reaction, which is sixth reaction. In this reaction, glyceraldehyde 3-phosphate, obviously two molecules of glyceraldehyde 3-phosphate will be converted into two molecules of 1,3-bisphosphoglycerate. So in this again, one phosphate is getting attached in the first carbon of that molecule. So first we are having phosphate in only third carbon. Now we are having 1 and 3. So hence it is named as 1,3-bisphosphoglycerate. And the enzyme which is taking part in this reaction is glyceraldehyde 3-phosphate dehydrogenase. So 2-pi, the inorganic phosphate, is coming into this reaction, which is getting attached in this first carbon position, and 2-NAD it is coming like taking part in this reaction, and it is released back into the cytosol as 2-NADH plus H plus. So NADH will be also counted as an energy, because as we read further we will get to know that 1 NADH is equal to 2.5 ATP or 3 ATP depend. So that we will be discussing later. So now so this reaction is a oxidation and phosphorylation reaction. So next so from this sixth reaction, everything whichever like from substrate to product, everything will be 2-2. So like which I have mentioned in my diagram, you can see there. So in next reaction, seventh reaction, this 1,3-bisphosphoglycerate will be converted back into 3-phosphoglycerate, two molecules of 3-phosphoglycerate, and with the help of the enzyme phosphoglycerate kinase. So you can see that phosphate which was attached to the first carbon, it is released back as 2 ATP. So this phosphoglycerate kinase, what it is doing, it is taking the first carbon phosphate group and it is attaching it to 2 ATP. As a result, 2 ATP is formation is happening there, and that 2 ATP it is released back to the cytosol. So till now we have formed 2 ATP successfully. Now we will be coming to the next reaction number 8. So in this reaction, this 3-phosphoglycerate will be converted into 2-phosphoglycerate with the help of the enzyme phosphoglycerate mutase. Mutase usually what it does, it changes the position of phosphate. Like if it is in first carbon, it will change it to in second or third, like that. So this 3-phosphoglycerate is converted into 2-phosphoglycerate. This reaction has some importance which will help in the further reactions to go on. So next, this 2-phosphoglycerate will be converted into phosphoenol pyruvate with the help of the enzyme enolase. So enolase, what it will do, it will generate one H2O, and which will go out from this 2-phosphoglycerate. So without the OH group of carbon 3 and H group of carbon 2 will generate one H2O, and that will leave. So we have this final product which is named as phosphoenol pyruvate. This phosphoenol pyruvate, it will come to the last reaction where pyruvate kinase will convert this phosphoenol pyruvate into pyruvate. So phosphoenol pyruvate, if you look into this structure, in the second carbon it is having one phosphate group. So again 2 ADP will come. This pyruvate kinase will take this phosphate group from the second carbon and give it to the ADP, so which will generate 2 ATP as a result, and this phosphoenol pyruvate will be converted into pyruvate. So now we have generated total 4 ATP. So that is, and this is the second ATP forming reaction. And one more thing, the 7th reaction, in which is the first ATP forming reaction, and this 10th reaction, both are substrate level phosphorylation reaction. So while discussing it in detail, I will tell you what is substrate level phosphorylation. So this is our whole glycolysis pathway. Now we will talk about some important points which we need to remember, which will help us in our national exams like GATE and NET, and which is like very common questions comes from these facts. So first is addition of phosphate to ATP or hexo sugar is always done by the kinase enzyme family. As you can see in the first step, as you can see, hexokinase, what this enzyme has done, this has taken phosphate from the ATP and it has given to the glucose, so it generated glucose 6-phosphate. And like that also in third step you can see phosphofructokinase, again kinase is there, so it is taking one more phosphate from the ATP and it gave to the fructose and converted it into fructose 1,6-bisphosphate. And in 7 and 10 reaction, you can see again 2 kinase family enzymes are there. In 7 it is phosphoglycerate kinase, which is helping to generate ATPs, and in 10th reaction it is pyruvate kinase, which is generating the final ATP. Next is pacemaker step of glycolysis is the third step. Always remember, the third step of the glycolysis is the pacemaker step of glycolysis, which is actually helpful to regulate this glycolysis pathway. So in this step, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Next point is priming reactions. There are 2 priming reactions in the whole glycolysis pathway. First one is glucose to glucose 6-phosphate conversion, and the second is the third step in which fructose 6-phosphate is converted into the fructose 1,6-bisphosphate. Next point is NADH formation. If you look into this glycolysis pathway, you will see this NADH formation happens in the step number 6, where glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. This reaction is an oxidation phosphorylation reaction. Next one is substrate level phosphorylation. There are total 2 substrate level phosphorylation reaction is there in this whole glycolysis pathway. First one is step number 7, where 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, and step number 10, where phosphoenol pyruvate is converted to pyruvate. So overall ATP is invested 2 ATPs, and ATP gained, it is 4 ATP, and NADH gained 2 ATP, and 1 H2O molecule is generated. So this is some important points of glycolysis. So that's it for today, guys. I hope you liked my video. I hope you liked the way I taught you. It was my very first video. So don't forget to leave a comment like how you liked the video, what are the changes you want to see in my future videos, and how I can improve myself. Definitely you should leave a comment like that. And if you like my channel, don't forget to subscribe, don't forget to like and share with your friends. Thank you. See you again in my next video.